Hello, I am Arnaud Delobel, R&D Director at Quality Assistance. Today I'm going to walk you through our comprehensive offer in mass spectrometry and protein characterization. More details are coming up in the next part of the video, where I'll be presenting slides over the services we developed at Quality Assistance and our expertise. Well, the characterization of proteins by mass spec is not something new at Quality Assistance. We started providing such services to our customers in 2006 when we installed our first high-resolution mass spectrometer, a Q-TOF Premier from Waters, and then a MALDI-TOF and an ion trap from Bruker. With these three mass spectrometers, we were able to provide a wide range of services, including intact mass analysis, peptide mapping, or glycosylation analysis. We were also equipped with several triple quads for quantitative LC-MS/MS. Since 2016, we have upgraded our mass spectrometers to offer state-of-the-art services in a full GXP-compliant environment. Today, we are equipped with 3 Xevo J2XS Q-TOF controlled by Unify. H-class Bio UHPLC systems are coupled to those systems, and 1 Xevo is also equipped with a multiple earth-cut 2-dimensional liquid chromatography and an HDX2 integrated system that you can see here. Except for HDX analysis, all those systems are controlled with Unify to guarantee full data integrity and GXP compliance from data acquisition to reporting in a single software solution. Recently, we have also implemented a bioaccord system from Waters for routine characterization of biotherapeutics. Thanks to its ease of use, its robustness and its lab-to-lab reproducibility, transferring characterization methods to QC is now much easier. To answer more specific questions, we also have a QDA mass detector, a single quad for routine analysis, but also an Amazon ion trap that is able to perform ETD fragmentation, which can be useful for the analysis of challenging post-translational modifications, a MALDI-TOF, and several Xevo TQ and TQS triple quad systems for quantitative analysis. All these systems are integrated in our quality system, and we can provide those services in a full GXP environment.
In the last few years, we have developed a set of analytical workflows for the characterization of biotherapeutics with a strong focus on monoclonal antibodies, ADCs, antibody-drug conjugates, and fusion proteins. These methods can be used as generic methods in some cases, but most of the time we use them as a starting point for the development of product-specific methods. The first workflow is dedicated to the analysis of intact proteins and subunits by 1D or 2D LC-MS. Intact mass analysis of monoclonal antibodies with or without prior glycosylation allows for a very quick characterization which can provide useful information on the identification of the antibody based on its molecular mass, but also on the glycosylation profile. Even if it won't be possible to identify small glycoforms, you will get data to compare batches and check their quality without any sample preparation, as this whole thing is performed online on a reverse phase column. In the case of antibody-drug conjugates, intact mass analysis also allows for the determination of a critical quality attribute of ADCs, the drug-to-antibody ratio, or DAR, and the drug distribution profile. This can be done very easily using the same method as the one that is used for MABs, except in the case of interchain cysteine-linked ADCs, such as Adcetris, one of the ADCs currently approved by the EMA and the FDA. In this case, a so-called native analysis is required in order to maintain the non-covalent interactions between the different chains. The raw electrospray Q-TOF mass spectrum is shown on the slide, together with a deconvoluted mass spectrum. The different species containing between 0 and 8 drugs are detected, and based on their relative intensity, the mean DAR value can be calculated. In order to get a better resolution and a better mass accuracy, the analysis can be performed at the subunit level. The antibody can be reduced to heavy and light chains, but it is much more efficient to use an enzyme called IDS that cleaves the antibody scaffold below the end region to yield 2 fragments. After further reduction, 3 fragments are obtained, FC, light chain, and FD, that have masses around 25 kilodaltons. With such molecular masses, very high mass accuracy can be obtained, and with the right choice of LC column and mobile phases, a nice chromatographic resolution between variants, such as oxidized species or lysine clipping, can be obtained, which allows for an accurate quantification based on the UV spectrum. The subunit approach is also useful for ADCs, as shown here for Adcitris, a cysteine link conjugate. The different fragments, with or without drug, are well separated, and by combining UV detection and MS identification, the average DAR value can be obtained accurately. With this analysis, additional attributes can also be characterized, such as glycosylation or post-translational modifications. Our Xevo J2XS cutoff is also coupled to a multiple hard-cut two-dimensional LC system. To show only an example of what it can be used for monoclonal antibodies, you can see on the slide the identification of charge variants of adalimumab. Ion exchange chromatography is not directly compatible with MS due to high concentrations of non-volatile sites in the mobile phase. Instead of collecting fractions and analyzing them by LC-MS, it is possible to use a 2D system with an ion exchange at the first dimension and reverse phase in a second dimension for online desalting and, if necessary, separation of species that co-elute in the first dimension. Here, we were able to identify at the intact level deamidated species, oxidized species, and lysine clipping variants. It can also be very useful for ADCs. You can see here the separation of variants of Adcitris by hydrophobic interaction chromatography, or HIC. HIC requires the use of mobile phases with a very high concentration of salts and is not compatible with MS. With multiple hard-cut to DLC, we were able to identify the two isomers of the DAR4 species, with the drugs being linked either between the heavy and light chains or between the two heavy chains. Let's move now to peptide mapping, which is probably the most used methodology for the characterization of therapeutic proteins. The main goal of this analysis is to confirm the identity of a protein by analyzing the peptide mixture obtained by enzymatic digestion, usually a digestion with trypsin. With UHPLC columns with sub-2 micron particles, an excellent separation between the peptides can be obtained. Until recently, most QC methods only used UV detection to compare the chromatograms of a sample and a reference standard. But now, MS systems can be integrated quite easily in a routine environment to add mass data to the analysis. With additional MS data, you have access to the identity of all the peaks, including new peaks that may arise with a new manufacturing process or during a stability study. You are able to identify and quantify low abundance variants, such as deamidated or oxidized species, that may not be resolved from the parent peptide. And with fragmentation data, you can sequence the peptides to have a better confidence in the identification and to sequence N and C-terminal peptides. In the case of ADCs, peptide mapping can be used to get the same information as for MABs, but it is also possible to specifically identify conjugated peptides. On this slide, the bottom chromatogram shows all the conjugated peptides of Kadcyla, a lysine-linked ADC. This information is useful for batch-batch comparison or for fine characterization in early stages of development. Another topic on which we dedicated substantial efforts is the analysis of glycosylation. Glycosylation is commonly considered as a critical quality attribute of therapeutic proteins and more specifically of monoclonal antibodies, as it has a strong impact on both safety and efficacy of the drug. We developed several analytical workflows, as shown on this slide, that we published in Journal of Chromatography A. Depending on the level of details that is required, on the type of glycosylation present on the protein, you may choose to analyze the protein at the intact or subunit level to get a global view of a glycosylation pattern. But you can also work at the glycopeptide level to perform site-specific glycosylation profiling. All these techniques require mass spectrometry, either on a Q-TOF or on a MALDI-TOF, and the combination of liquid chromatography separations by reverse phase, HELIC or PGC, and sometimes even mixed mode chromatography. But the most common technique that is used for routine testing of monoclonal antibodies, for instance, is N-glycans profiling after PNGA's release of glycans and derivatization, usually with 2-aminobenzamide, 2AB, or more recently with rapid fluoro-MS. This methodology was used in the past with fluorescence detection, but now we use routinely MS in addition to fluorescence, which allows for an automatic identification of the different glycans within Unify software. Using rapid fluor MS labeling, it is easy to identify and quantify glycans below 0.1% of relative intensity. And finally, I will tell a few words about HDX-MS. HDX stands for hydrogen deuterium exchange. The principle of the technique is quite simple. We will incubate a sample in the presence of D2O, water in which hydrogen atoms are replaced with deuterium. Hydrogen atoms of the peptide backbone, amide hydrogen especially, will be exchanged with deuterium at a rate that is dependent on several parameters, one of which is the accessibility to the solvent. The exchange will be quicker for hydrogen atoms that are exposed to the solvent and slower for atoms that are buried in the inside of the protein. HDX experiments can be very time-consuming, but systems such as the HDX2 system can perform the analysis in a fully automated way to improve the precision of the method. We are equipped with such a system that includes an Acuity M-class system for the chromatographic separation and an online pepsin column. Everything is done automatically and coupled to our Oxevo J2XS QTOF. Here is an example of a structural comparison between a stressed antibody and the reference sample that highlights structural modifications in several regions of the antibody, especially in one of the CDRs, which may have a significant impact on binding properties of the antibody. If you want more details on the technique, I recommend you have a look to our dedicated webinar on our website. As you can see, we have a wide range of services already available for our customers, but we keep improving those services and several developments are in progress in our lab. First of all, multiple attribute monitoring. In this case, the goal is to use a single LC-MS method based on peptide mapping in order to not only identify the protein, but also quantify post-translational modifications such as deamidation or oxidation, characterize the glycosylation profile, detect fragments, and quantify process-related impurities in a single analytical run. A second project is the identification and quantification of host cell proteins, HCPs, by mass spectrometry using a proteomics approach. We will also improve our workflows in 2D LC-MS, for example, for the characterization of in-process samples without any sample preparation, or for automated subunit analysis. And finally, we will implement cross-linking mass spectrometry for the analysis of higher-order structures. All those MS characterization services are a part of the techniques we provide for biotherapeutics. Our services cover everything from physicochemical methods to cell-based assays for both CMC and bioanalysis. All the studies are tailored to your own needs depending on the stage of development of your products. And all our laboratories are located on one site in Donstienne, Belgium. If you want to learn more about MS characterization at Quality Assistance, you can access many application notes on the scientific library section of our website. Several publications are also available and more will come in the next few months. All the links are available on the website. And finally, webinars are also available on demand that cover all the workflows that I have presented today. Please don't hesitate to contact me if you have any question, and see you soon.